Welcome to Computational Biology. Today, we are diving into the fascinating world of small molecule databases. If you have ever wondered how scientists discover life-saving drugs so quickly in the modern era, you are in the right place. We're going to build your understanding from the ground up, starting with a simple biological analogy and moving all the way through the mathematics of how we search for new medicines. Let us get started. To understand why these databases exist, we first need to understand the biological problem they solve. Imagine you are a detective trying to find a specific key for a very complex lock. In the human body, diseases are often caused by malfunctioning proteins, which act exactly like these complex locks. To fix the problem, scientists need to find a tiny chemical key that fits perfectly into the protein to turn it off or on. Instead of testing every single chemical in a physical laboratory, which takes years, researchers use computers to search through massive digital hardware stores. Now that we understand the lock and key analogy, let us define what these digital hardware stores actually are. In computational biology, a small molecule database is a highly structured digital repository. It stores comprehensive information about low molecular weight organic compounds. When we say small, we specifically mean compounds with a molecular weight of less than 900 daltons. This specific size limit is crucial because it allows these molecules to easily cross cell membranes and interact with the biological macromolecules, or the locks, inside our bodies. Knowing what these molecules are leads us to ask what exactly we store about them. These databases contain much more than just a simple list of names. They store the precise two-dimensional and three-dimensional chemical structures of these molecules. But structure alone is not enough. The databases also catalog their physical properties, their known biological activities, and their potential toxicity. Having all this data in one place allows scientists to predict how a molecule might behave in the human body before a single chemical is ever synthesized in the lab. With all this data available, you might wonder where scientists actually go to find it. There are several major databases used globally. PubChem is one of the largest, housing millions of chemical structures. DrugBank provides highly detailed clinical and pharmacological data, specifically about approved drugs. Zinc is a specialized database used primarily for virtual screening of commercially available compounds. Finally, the Cambridge Structural Database stores experimentally determined three-dimensional crystal structures. Together, these databases form the foundational infrastructure for modern computer-aided drug design. Having these massive catalogs at our fingertips has completely revolutionized the pharmaceutical industry. Before these databases existed, discovering a new drug was a slow, expensive process of physical trial and error, often taking decades and billions of dollars. Today, if a new virus emerges, scientists can instantly download the three-dimensional structure of the viral proteins. They can then computationally screen millions of small molecules from databases like Zinc or PubChem to see which ones might block the virus. This rapid computational process is known as virtual screening. This brings us to an important technical question. How do computers actually search through millions of molecules so quickly? When searching a small molecule database, we rarely look for an exact match. Instead, we look for molecules that are structurally similar to a known drug or a desired template. To do this efficiently, computers convert complex chemical structures into simple binary strings called molecular fingerprints. In these strings, a 1 means a specific chemical feature is present, and a 0 means it is absent. This allows the computer to compare molecules mathematically. Now that we have our molecules represented as binary fingerprints, we need a way to calculate their similarity. Computational biologists use a mathematical formula known as the Tanimoto coefficient. Let us break down the symbols. T represents the Tanimoto similarity score between molecule A and molecule B, which always ranges from 0 to 1. The variable A represents the total number of chemical features present in molecule A, while B represents the total features in molecule B. Finally, C represents the intersection, meaning the number of chemical features that both molecules share. If they share no features, C is 0, and the score is 0. As shared features increase, the score pushes closer to 1, meaning identical fingerprints. Let us see this formula in action with a concrete example. Suppose we are comparing a known painkiller, molecule A, with a newly discovered compound, molecule B. We are given that molecule A has 50 features, molecule B has 40 features, and they share 30 features in common. We want to find their similarity score. We choose the Tanimoto coefficient because it perfectly handles these molecular fingerprints. We substitute 50 for A, 40 for B, and 30 for C into our formula. The denominator becomes 60, representing the total unique features. Dividing 30 by 60 gives us a final score of 0.5. In computational biology, a score of 0.5 means the molecules are moderately similar, warranting further investigation. Now that we have used the formula to find a score, let us tackle a more advanced problem. A researcher is screening the zinc database for a molecule similar to a known anti-cancer drug. The known drug has 120 features, and a candidate molecule has 100 features. To be considered a hit, the candidate must achieve a Tanimoto score of at least 0.80. We need to find the minimum number of shared features, C, required to pass this threshold. We start by setting up our equation, plugging in 120 for A, 100 for B, and 0.80 for the target score. This chunk of our derivation establishes the baseline equation we need to solve. Continuing from our setup, we now need to isolate the unknown variable C. First, we simplify the denominator by adding 120 and 100, which gives us 220 minus C. Next, we need to eliminate the fraction to make the algebra easier to handle. We do this by multiplying both sides of the equation by the denominator, 220 minus C. This mathematical reasoning brings the unknown variable C to the surface, allowing us to group our terms. We are now one step closer to finding the exact number of shared features required for our cancer drug candidate. In this final chunk of our derivation, we will solve for C completely. We distribute the 0.80 into the parentheses, giving us 176 minus 0.80 times C, all equal to C. Next, we group all terms containing C on one side by adding 0.80C to both sides, resulting in 176 equals 1.80C. Finally, dividing by 1.80 gives us approximately 97.78. Since a molecule cannot have a fraction of a feature, we round up. The candidate must share at least 98 features with the known drug to be considered a viable hit. So, we have proven how the mathematics works. This brings us to a visual summary of the entire process. On the screen, you are looking at a complete schematic of virtual screening. On the far left, we have a cylindrical database representing PubChem or zinc, filled with thousands of small molecule structures. An arrow points to a computer monitor in the center, which displays the binary molecular fingerprints we just learned about, comparing ones and zeros. Finally, an arrow leads to the right side, showing a large, complex blue protein structure, the lock, with a small molecule, the key, fitting perfectly into its binding pocket. This diagram beautifully illustrates how digital data is transformed into a physical biological solution. We have covered a lot of ground today, so let us reinforce the three most important ideas. First, small molecule databases are the digital backbone of modern pharmacology, cataloging millions of chemical structures to replace slow physical trial and error. Second, computers search these massive databases by converting chemical structures into binary molecular fingerprints. Third, we use mathematical tools like the Tanimoto coefficient to quantify chemical similarity, allowing us to rapidly identify promising drug candidates. You have now covered the full topic of small molecule databases, understanding both the biological concepts and the mathematical tools that drive modern drug discovery. Great job!